Great. Well, thanks, Jay. Welcome, everybody. I know some, but not all of you. I'm Corey Buxton, and I'm a professor in the College of Education here at Oregon State. And two other members of our team, Deanna Crespo and Barbara Etnauer, are joining us as well, and they'll be in the breakout rooms. I was super excited because this request actually came from the Smile teacher community wanting some lessons developed on vaccines, the role of vaccines in our communities, infectious diseases. And while I am not an epidemiologist, like most of you, I've had to learn a lot of epidemiology in the past couple of years. And so I was excited to try to pull together some resources that I think will be relevant and meaningful for our students. But of course, you are the experts and need you to tell me and to tell us how we can sort of continue to massage these. We're hoping you'll try them out, give us some feedback, help us make them better. So I think lessons on topics like this are really important. Science, obviously, relevance in our lives. We talk about the need for relevance. Nothing may be more relevant than keeping our families and communities safe. We know that science is one way of knowing. It's not the only way of knowing, but it is good for knowing some sorts of things. And public health is an area where science kind of has a long history of showing that it can be helpful. And so we want to, not like tell our students what to believe. We want to show them the evidence and let them come to their own conclusions. And I think that's how learning meaningfully happens. And so that's what these lessons are kind of about. So, and these are part of a project called Lacunas. And some of you who are in Smile, if you were at the August workshop, you've heard about Lacunas. And Lacunas stands for Language, Culture, and Knowledge Building through Science. It's an NSF-funded grant we have here currently at Oregon State. And it's building on, basically, I wrote a proposal that said, we know new things now about how people learn. We've learned a lot about learning in the past couple of decades. We know it isn't just cognitive, it's also affective, right? We know we learn best when we feel safe, when learning feels relevant, when we feel engaged in what we're learning about. And we know that some of the work that has been done with special populations, English learners, students with special needs, that has taught us a lot about how all students learn. And so this project is really about using some of what new things we know about how kids learn to hopefully make science more meaningful and approachable to them. And so within Lacunas, we talk about kind of three areas. We talk a lot about the role of language and how we need to think explicitly about language and make our thinking about the language of science more conscious and help make this understandable for students. We have to talk about language and how we use language to make meaning. We also talk a lot about community connections and cultural connections. How do we support students' interests and identities? We have to make science relevant to them, right? And finally, we want to talk about how we build knowledge over time and how we come to believe what we believe. Right? Everybody comes to conclusions about things. What do we use as evidence to shape our decision-making? And how can we make good decisions using the best evidence we have? So those are some of our kind of overarching goals in Lacunas. And we use these model lessons that embed some tools and practices as a way to try to make these things come to the surface. So before most of our time today, I really want to focus on the two model lessons. I just wanted to say a couple of things about using socioscientific issues. So this is not a new idea in science ed. People have been doing this for at least a few decades, thinking about what are the issues in our communities that are social and scientific and why these might matter particularly to kids and families and might get people interested in talking about science. And we know as teachers that connecting new concepts to students' prior knowledge is really important, and connecting their out-of-school lives to their in-school learning is really important. We know this, right? This is not news, but it's hard, and we may struggle to do it consistently and strategically. And so SSIs, these kind of socio-scientific issues, potentially are a good way to bring this relevance really front and center, topics that kids really care about: climate change, climate anxiety, vaccines, disease, birth control. There's a whole lot of issues. They're touchy. They can lead to conflict, but these are the real life issues that, young adults need to make decisions about, right? We're not doing them any favors by staying away from sensitive topics because they live in a world where these things are everywhere and they have to make decisions, right? And so we're going to use two model lessons about the topic of infectious diseases and vaccines to highlight maybe how we do this, how we make some of these topics relevant, and how we put evidence in front of our students and let them do some of the work of coming to their own decisions. Okay, so we're going to go into our first lesson. And so I was asked to come up with a lesson that sort of talked about the vaccine and thinking about getting vaccinated, but I recognized, as most of you probably have if you've talked about these topics with your students, that we have to take a step back, and before we can really start talking about vaccines, we have to talk about how infectious diseases spread in general, because vaccines only make sense within a context of, okay, there's a disease spreading and there's multiple things we can do to try to keep our families and our communities safe, right? So we developed a first lesson that's more on diseases spreading in general, and then the second lesson we'll look at is looking more explicitly at the role of vaccines and other ways to mitigate spread. Okay, so we are going to now leave our PowerPoint and hop over to a Word document. And this is kind of the student packet, the student handout on how infectious diseases spread. And so we'll walk through this. And for those of you who maybe are new to Lacunas, I'm going to point out a couple of the features that we build into all our lessons to support these three strands of language development, community connections, and knowledge building. So we always start with what we call a language booster, and a language booster is it's a couple paragraphs. It hopefully is a hook for students, and it's connected to the idea. It may be a little tangential, but it's a way in for students. It introduces some of the core ideas, some key vocabulary, some key concepts, and then asks students to do some thinking. So the language booster for this lesson is about the cholera epidemics in London in the 1800s. And that's important because that is sort of the history of epidemiology. That's the birth of epidemiology, was a medical doctor named John Snow, who came up with a way to sort of test his hypothesis that cholera was a waterborne disease when most of the doctors at the time thought it was airborne. And he didn't have evidence to show that it was waterborne, so he had to collect data, and eventually he got the city to take the handle off a well pump and show that the disease stopped spreading when the well was no longer in use. So it's a language booster to engage kids in some of these ideas about epidemiology and introduce some history, and hopefully is an interesting story. Scroll down a little bit, Jay. You'll also notice some words in bold. Those have concept cards at the back, which we'll show you at the end, and we'll talk about that a bit. So the language booster is short, and then it ends with a couple questions, and I think they're on the next page. But this gives you a sense that in Lacunas, the types of questions we want students to sort of talk about are, when we think about language, it's to actually sort of think about how we use language and how we can change language to make ourselves understood better. So a question like, choose a sentence and rewrite it in a way so a second grader could understand it, right? It's not a reading comprehension question, but it's a question about language. Write a sentence that describes in your own words the evidence that Dr. Snow used. Now that's a reading comprehension question, but it's asking students to rework things and not just copy an answer, right? So that's just an example of how we try in our questions to ask questions in a way that is always asking kids to do things with language. And it's just sort of an aside. But the meat of this activity is two simulations. So there's a computer simulation, and then there's a hands-on simulation. And I think the combination of the two really help to make some important points about how diseases spread. And one other side note I should say is we sort of think about these as being targeted at middle school. So for middle school teachers, you can tell us if you think we hit that right. If you're high school, you might think about how you might want to add to it. And if you're elementary, I'll be curious to hear if you think that some of this is feasible they just need a little bit of extra support. So we go from the language booster into the actual investigation. In this case, our investigation is two simulations. And a really important concept here, excuse me, in that diagram, we also try to put in a lot of pictures and figures and labeled diagrams. We think that multimodal is really important for all learners to see information in different ways, right? So reproduction number is a really important concept, and it's basically simply if one person is infected with an infectious disease, on average, how many people will they spread it to? That's the reproduction number. So if a disease has a reproduction number of 2, every infected person is going to infect 2 other people. So that's what this model shows, a doubling. Each one person makes 2 other people sick. So that's a reproduction number of 2. And if something is spreading with a reproduction number of 2, it's going to get out of hand pretty quickly. And if the number is higher than 2, it's going to get out of hand really fast. If the number is between 1 and 2, it will keep growing, but more slowly. And in order to not have a disease spread, we've got to get the reproduction number down to 1, which is basically a flat line, or less than 1, where it will start to decline. So that's an abstract concept, right? It sort of makes sense to us, but to our kids, pretty abstract. So we need some simulations to help us see this. So the first simulation is basically just a Google spreadsheet that has been pre-programmed, and it asks students in groups to basically put in 3 variables. So we can talk a little bit about what are some of the features or variables that might affect the reproduction number. And so we need to think about how many people are originally infected. That's going to affect it. And then what our reproduction number is. So how volatile is it spread? And then we want to model a certain number of rounds of infection. Of course, in the real world, it doesn't really work in rounds, but you could think about it as like exposures, number of times you come in contact with someone who's potentially exposed. So in this case, you could start with 1 person infected. And if we put a reproduction number of 1, then it doesn't matter how many rounds of infection. So just looking at the blue, so there's a blue and a red, and they'll show up as lines then on the graph. So you can see if we've got, for blue, 1 individual and a reproduction number of 1, it's always going to stay 1. It's just going to be a flat line, 1, 1, 1, 1, 1. And if there was a reproduction number of 0, similarly, it's obviously not going to spread, 0, 0, 0, 0, 0. So the fun comes with changing some of those conditions, right? So if we change situation 1, you know, it's interesting to think about number of individuals. So one of the questions is, which of these is going to affect the number of infected people most? So there's some good conversation you might want to have with students before you let them start plugging in numbers, just to think about what they might see. But then just give them some time to put in numbers and play around with it and just see what happens. And then there's a little bit more guidance. But so clearly what you'll see is as the reproduction number goes up, you'll start to see a curve that starts to look exponential, and you can see it both sort of numerically in the column and then graphically. And you can just keep this with 1 column, that's fine, or you can play with 2 different conditions to compare. So keeping 2 numbers the same and then changing the 3rd variable is a good way to suggest that students do this, just to look at the effect of if you change individuals initially affected, that actually isn't going to make that big a difference. If it's 1 or 10 initially, what matters way more is the reproduction number and the number of rounds. So when the reproduction number goes up to 3, you can see people start getting infected really exponentially, and it looks very nasty. So this is hopefully a pretty straightforward simulation. There's not a lot that you can do with it, just changing those 3 variables, but you can, with comparing 2 cases and having 3 variables, you can actually talk about a lot of topics, and the goal would be to get the students to just sort of have a conversation about what surprises you, what did you expect, and what actually happened. Kids are often surprised how quickly, with the reproduction number of 3, after 8 rounds of exposure, you've got 65,000 infected people. And of course, in the real world, it wouldn't work exactly like that, but it can be pretty striking that if we don't do something, the takeaway ought to be, if we don't do anything and these things spread, they pretty quickly can get quite out of hand. And that's the computer.
Sorry to interrupt. There was a question in the chat. Could we maybe go back to the Excel sheet? And there was a question about what happens in situation 2.
Yes, so simulation 2 is just like simulation 1. You can just have a second case. So basically it allows you to compare if we started with 10 people in each case, and in one virus, the reproduction number is 1, and in another virus, the reproduction number is 2, you can see the effect of changing the reproduction number from 1 to 2, or in this case, from 3-1 or whatever, 3-5, and still 10 rounds. So it's just allowing you to compare two cases. If it's simpler for your students, you can just limit it to one, and that shows you that case. Does that help?
If you go down that spreadsheet, there is some direction that you can use, like those instructions back down there. Yeah, those. They are really interested to do if COVID is 1.3 and flu is 1. So that's are interesting also to try.
Yeah, so there are some suggestions there for things you can try. And we put some of that when we go back to the lesson. A few of those are built into questions at the end. Okay, so that's the first simulation. And so that's a computer simulation. And then students might have, all kinds of thoughts and questions about that. Those are just sort of the directions of what we went through. And if you scroll down just a bit more, Jay, on the next page, then these are some questions that are grounded basically in those questions that were down the bottom of the spreadsheet, but it's just allowing students to reflect a little bit on what they saw in the simulation. But simulations and computer models, I find with kids, they can make an impact, but they're not quite as powerful sometimes as like a hands-on demonstration. So we also have a parallel hands-on simulation of how infectious diseases spread. And so for this one, basically, kids just have a little test tube and a dropper. And for everybody but one student, it's just distilled water. So we'll send you materials and just little plastic test tubes and droppers and some distilled water, and then a pH indicator, probably sodium hydroxide, low molarity sodium hydroxide to do a pH test. So basically, you're as the teacher, you're gonna set up one student as patient 0. So everybody is going to get a regular distilled water tube, except one person that you're going to give the tube to who has the distilled water plus NaOH. And so obviously that tube has a different pH. And so it's basically just using that pH indicator to spread the quote unquote infectious disease around the room. So the way the procedure works is you get your students to just kind of mill around and then you say stop. And then you're going to exchange liquid with a partner. So you'll take your dropper and take some of your liquid and give it into your partner's test tube. They'll stir it around and then they'll give you some of theirs back and you'll mix it. So basically, patient 0 who had a vial, with a pH of 10, they're going to spread that in the first round to one other person. So there'll be 2 people, then they're infected. And then you say, okay, move around again, and we're going to share again. So those 2 people should probably infect 2 other people. And then you do a 3rd round and share. And so probably, depending on how many students you have, typically about half of the class actually ends up infected. So then you have people go back to their seats and you come around with some phenolphthalein, pH indicator, and test everybody's tube. And anybody who got infected, theirs is going to turn pink because they've got a different pH. And if they never shared with someone who was infected, then obviously they won't turn and okay, you're not infected. So then there is this is the work of epidemiologists. We have some infected people and some non-infected people. And I'm telling you that somebody was patient 0. One person started with the disease. And the job of the class then is to try to figure out who was patient 0. And so it'd be interesting to sort of brainstorm a bit with y'all about how to do that, but we don't really have time. But in effect, what they should come up with is some sort of tree diagram. And basically, you've got to look at if I'm not infected, then anybody who I shared with wasn't infected when they shared with me, right? Or I'd be infected because this has 100% spread rate. So if I shared with somebody infected, I'm infected. So if I'm not infected, then either nobody I shared with was ever infected or they got infected after they shared with me. So the interesting case is I shared with Tim and then at the end, Tim is infected, but I'm not. How is that possible? Well, it means that Tim got infected not by me, but by somebody else he shared with after he shared with me. And so there's sort of this puzzle that you have to sort of trace backwards, and it's helpful to draw kind of diagrams, and people come up with different strategies, but the idea is for the students to try to think about and represent how would you try to figure out who was patient zero. And I found that the combination of this kind of hands-on simulation combined with computer simulation really can be pretty impactful in getting students to go, this stuff can be it can spread pretty fast and exponentially in a way that seems kind of scary. And that really is sort of the takeaway from this lesson is without doing anything to control spread, these things can really get out of hand. And we don't want that. We want to keep our communities safe. So that is sort of this first activity. And then the only other things, if you keep scrolling, Jay, a bit, we always have kind of a summary page that asks students to think about what they learned. So in this case, how can simulations help us explain or understand how diseases spread? But then we also like to ask questions that get students to think about the language they would use to explain this. So in your own words, how would you describe what you did? And then how might an epidemiologist, now you've learned some language that epidemiologists might use, how might they describe what you all did to try to figure out who was patient zero? And the very last thing in the lesson then are the concept cards. I said that we introduced some bold words in the beginning, and we make concept cards that are bilingual, that have kid-friendly definitions, that are multimodal, that have images, that use the word in a sentence, and all in all are trying to make these words familiar, useful, and usable to our students so they can start to talk about epidemiology and patient zero and reproduction number. And that can be helpful, not that you need those words to understand these ideas, but when we can use this language, it helps us to be convincing. And part of what this is about to me is learning, like, what do I believe we ought to be doing? And then how do I convince other people that this is what we should be doing, right? And so using this language and using these ideas can help us to be clear about sort of why we think we understand things the way we do. So we are going to hop into breakout rooms for a few minutes now, and the goal is to think about this lesson, to share any insights. I'm particularly interested in any thoughts about the language and how we support that, and the connections to the community and how we do that. But really, we're just also very interested in your feedback on, would this lesson work for you? What might we do to make it better? And just what thoughts do you have about using this lesson? Great conversations in our group, I'm sure in the other group too. Unfortunately, in a 1 hour PD with 2 lessons, we're just going to keep rolling along. So hopefully, we captured some of the ideas in the Google Doc. Okay, so the first lesson was about spreading infectious diseases when there are not things being done to try to mitigate the spread. So the second lesson is on things we can do to mitigate the spread of a disease like COVID or influenza. And so this lesson, again, sort of has 2 models. Unfortunately, there's not a cool hands-on simulation for this one. There is a computer model, and then there's like a diagram basically for students to look at and think about. Vaccines lesson, okay, so very parallel. This lesson, of course, also starts with a language booster. In this case, the language booster is a little bit of background on viruses, and particularly spike protein viruses like COVID, and how they trick our cells into sneaking in. So the first paragraph is just sort of that summary of how COVID gets into our cells, and hopefully a picture that helps students to see what that looks like. And then the other 2 paragraphs talk about vaccines and how different, there are different types of vaccines, and super interesting for people who geek out on this stuff, how we have the mRNA vaccines now. So, I mentioned it without going into any detail, but the idea that there's different kinds of vaccines, but basically they all work in parallel ways to teach our body how to resist a disease like this. And you could talk about something like the Black Death in Europe, and how when there was no medicine and there was no immunity, a disease just spreads and spreads and spreads and spreads, right? And so luckily we understand more than they did in the 1300s, and we've got some other tools. And so we have these vaccines as well as other tools that can help us. So that's the language booster, talks about viruses and vaccines. And then we've always got sort of a question or two. In this case, write a sentence, draw a picture to explain to someone in your family how the virus tricks your cells into letting it pass through. So hopefully they don't just copy the picture up above, but try to think about how they would represent what actually is happening when we get sick from a disease like COVID. So that's the language booster. And then we have the investigation. And again, this investigation has two simulations. The first one is a computer model that's about herd immunity. And so students might have heard of herd immunity, but maybe don't know exactly what it means. So there's a little bit of discussion about this idea of herd immunity, and that we can, diseases will spread less when either people are vaccinated or they have other immunities, such as they've been infected before, right? And so this is getting at this idea that a disease won't always have the same reproduction number in every community, in every context, because the context matters and affects how much it's going to spread. And so a disease is going to spread more in some communities than others based on a number of factors. So this is a computer simulation that basically compares, and this initial case compares 3 communities with different vaccination rates: 5% vaccinated, 30% vaccinated, and 75% vaccinated. And the little hexes represent 3 situations. Healthy people are the light gray, sick, infected people are the orange, and vaccinated people are the dark gray. And this is a true simulation in that if you run it multiple times, it's actually doing probabilities, and it will spread differently. And you've got for each one how many people are infected and how many are healthy. And one thing that students will see is if you run the simulation repeatedly, there will be patterns, but it won't be identical. The number of healthy people at the end will be different each time. So that's one thing worth noting. So maybe go ahead and run it, and you can see how it spreads. And not surprisingly, in the left case, when there's only 5% vaccinated, it spreads a lot and most people get infected. In the right-hand case, where most people are vaccinated, only 22 people got infected because infected people kept bumping in basically to vaccinated people and slowed the spread. And the middle case is kind of the middle. But if we run it again, we'll probably get different numbers. Instead of 22 infected people on the right, now there's 26. So the numbers are similar, but not identical. And if you run it enough times, like all bell curves, you'll get some cases where it ends up being pretty different, but the 3 cases will always be pretty distinct, that you will always have more spread in the unvaccinated, virtually unvaccinated community than the vaccinated community. So there's plenty you can think about with that. But one thing I like about this is if you scroll down, Jay, there's a couple other versions. So this first one is meant to model the Delta variant, where there's breakthrough cases. So even vaccinated people may get sick. And so if something is more infectious, or the vaccines or herd immunity is not strong, even communities that feel like they're safe may be slightly safer, but not much safer than communities with less vaccination. So that's one scenario you can talk about. Vaccines aren't perfect, vaccinated people still get sick. If you keep scrolling down, then we've got a case where now we've got the blue hexes, and those represent people that have been sick and basically survived and have some immunity. So much like they behave much like vaccinated people, right? So if you have a community where there has been a high impact already, you can have lower vaccination and still have some closer to herd immunity. Not the way you want to develop. Any epidemiologist will tell you want herd immunity through vaccine and not through people surviving getting sick. But people survive getting sick, and they also have some immunity, and that still helps communities stay safe. So that's just further sort of complicating the scenario. And I think there's even one more model down there where basically there is some initial immunity, but less than in the other case. And so this is sort of an intermediate case. So anyway, there's a lot you can see by playing around with these different versions of the simulation, and they vary, but hopefully the takeaway is you have to do something to build up that immunity if you want diseases to not spread like crazy around your community. Of course, like any simulation, like this is a model, this isn't the real world. So anybody, your kids, anybody could question this. Like, do I believe this? Well, you know, I have to look at the source. I have to look at where it comes from. I have to look at what if other people have said things about it. But no, and Ken Lacunas did not make these simulations. This one comes from National Public Radio. And the first simulation we looked at comes from a team at the University of North Carolina that makes simulations. So no, we are not responsible directly for either of these. So then there are some, that's the procedure for what you do, and then there's the link and the figure, the procedure, and then again, some questions for students in groups to think about, and thinking about what happened to vaccinated people, unvaccinated people, and then questions that the simulation raises. And then the last question is, okay, so in addition to vaccines, what other strategies have communities used to slow the spread of COVID or other infectious diseases? And that brings us to the second model, which is called the Swiss cheese model. And this is just, we'll send you basically like color laminated like handouts of this picture. But it's the idea that much like Swiss cheese with holes in it, any single approach we use to try to slow an infectious disease is only partially effective. And the more different layers of protection we can build in for a community or a family, the safer we should feel. And so nothing is going to be completely foolproof. The holes could line up and you could still get sick, but the more layers of defense we build in our communities, the less likely diseases will spread rapidly. And so we can talk about each of these pieces of cheese represents a different strategy, and some of them are personal and some of them are community actions. And I think that's the other thing I would really want to emphasize here for students to discuss, that there's personal responsibility and there's social or community responsibility, and there's personal freedoms and there's social, contract. And so we get to make choices in our lives, but we also have to think about the implications of those choices for other people. And we're part of a community, and some things that we do individually affect the community, and some things the community does affects us, and that's being human and being social creatures. So there's lots of great food for thought in this and lots of room to disagree about the effectiveness of different strategies, but I think framed as a conversation about all communities want to be safe, all families want to keep their family members safe, and what are the different tools we have, and we make choices about which ones of these we use. But there's lots of tools out there, and we should be thinking about how we use these tools to keep ourselves safe. So that's the second model. We're getting a little bit behind on time. Sorry about that. I'm talking too much. But yeah, the procedure to do this, and then noticing and wondering is just how we framed it. Look at the model, talk about it, what you notice, what do you wonder? What questions do you have? And I think that's kind of a safe way, maybe, rather than like, these are things you have to do. No, these are things that communities are doing, and let's talk about these and what they might cause. So that's what they do. And then the wrap up, rather than our normal like lab notes form, we're hoping that some of you might be willing to have your students try to do a concept map on this. I think this is a really nice topic to think about, a concept map, to use some of these concepts from the concept cards and to try to think about how they're connected in our students' minds. We're big on concept maps because it aligns with our view of sort of language and knowledge building. And so hopefully some of your students might be willing to work in pairs and create a concept map, and we've got sort of a structure to walk them through. Some of you have done concept mapping with your kids before, I know, and some maybe haven't. Jay has a concept mapping activity he's done. And we wanted to do a real quick poll with you all to see your experience with concept maps and your kids' experience with concept maps. We have another lesson that's just about concept maps and not about these topics that Jay developed. It's just basically on parts of a plant. So students ought to have some familiarity with the topic, and it teaches them concept maps. So yeah, if you could take just a second.
Okay.
Awesome. Some comfort, and students have some comfort, and some folks would like to have some more training on concept maps. So that'd be great. We would love to do a PD on concept maps, what they're good for, and how you might make good use of them.
So I think we should move quickly to the next breakout room. And I put 5 minutes. 5 minutes is going to be half the time, but twice the fun. Everybody's going back to the same room.
Yep, time flies when you're having fun. We are just about out of time. I hope that these lessons seem useful to you and that you will take at least parts of them and try them out and give us feedback. We'll be revisiting these and some other related things at one of the things we'll do at our January in-person Smile teacher workshop, as we will be talking a little bit more about these. And hearing about if you've done some of these, we would love to know. And especially the concept map piece, we'll be talking more with you about that because this is kind of an ongoing piece we'd like to build in and get some sample concept maps from students to see how they're making sense of these ideas. Maybe should I turn it over to you, Jay, for final thoughts, or should I talk about the evaluation link? Or? So
I'll just let you know here, you can email me directly. Many of you know my email address, but you can also just email smileprogram@oregonstate.edu. If you want lesson materials, we're going to be preparing these lesson materials and sending them out so that you have an opportunity to use them. But we are asking that you request them just so we know how many kits to make. But we'll be working on getting those together and sending those out quickly. So please let me know. We will be having future PDs for Smile teachers that will be going on, and we'll keep those coming out in the newsletter. And I sent out some reminders yesterday. If anybody has questions about it, I'll be here after the program, and people can stay and ask me any questions they want.
Yep, and I'll be hanging around as well. And we are hoping that we can ask you to fill out a quick evaluation survey on the PD training, which can be about the lessons too, but particularly we want to continue to do these virtual PDs and we want them to be as useful as possible. So I don't know if Jason, if you want to say a quick word.
Yeah, this is just, hi everybody. It was great to watch. This is just a really quick way of providing feedback to sort of guide future professional development. It should take you less than 5 minutes. It's really fast. It's all online. I just pasted the survey, the quick survey down below. Click on it, take 5 minutes. It's totally anonymous. And again, it just takes your feedback back to Corey and Jay so that we can improve the offerings. So thank you.
Jay, anything else you want to say to wrap up?
Well, we have come to the final part of this, and we made it on time. Thank you all for your participation. Be looking for more virtual teacher professional development, and a couple quick Smile reminders since I may have like 30 seconds. We are going to be having a winter teacher workshop this year in person at OSU, January 28th, 29th, 2022. Get your subs reserved now. And we'll be having more information coming on that soon. I am still looking to have community conversations with folks. There is a link in either the email I sent yesterday or the newsletters. You can set up a time with me. If one of those times doesn't work, just email me. We'll set up a different time. But I'm trying to talk with all communities, either as a whole community or just as individual teachers. Does not matter to me. But just trying to give you as much support as we can during what is a challenging year. So with that, thank you very much for your participation, and looking forward to seeing you online or in person very soon.